just press OK for that if it says recording. Okay, that's fine. Hello everybody. My apologies for being a little late starting. We just realised that somebody in the room was in the same department as I was, who had was taught by somebody else who supervised me, so it's quite exciting to get a connection from many years ago. Welcome to the cafe, particularly welcome to new people who haven't been before or haven't been for a very long time. It's lovely to see you. Welcome to our new place where we meet nowadays, in Pharmacology. Welcome to everybody online as well. A little housekeeping for the building. So if we did have a fire alarm, which I trust and hope we won't because there are not many students left in the building, then please make sure you go outside by the nearest stairs. I think the lifts will stop down the main stairs there, or push through the fire escape stairs, they go to the back, and then we'll meet over by the fence next to the horrible temporary buildings. That's actually that way. You will have come in past when you came walking in. Okay? So that's if something goes wrong. Hopefully it won't. Just to explain the way that the evening goes. Just can you turn off your audio just for a moment? Thanks, Peter. Perfect, thank you. We start with a little introduction and then a talk, and the aim of this is to kind of lay out the general area, but there will be many things I'm sure occur in people's heads. If you're online, you're very welcome to put comments, ideas, questions into the chat. Please keep yourself muted, and we will then use those and read them out later. And then if you're in the room, we're going to take a short break after Peter's talk, just long enough to refill wine glasses, soft drinks, take more snacks, because I don't want to take any wasted food home, otherwise I get fat. And then after that we'll have the discussion, which is open till probably about 8:40. We're a bit low on numbers of hands to clear up today, so I shall stop a little bit early. We are recording. This will then go out onto Zoom, on YouTube rather, later. For those who haven't used it before, we have private playlists for the cafe. If the speaker is happy to, I also put on the public one, which is then searchable. Thank you very much. So, I'm delighted to introduce tonight Peter Hore. Peter has actually been a long-term colleague of mine, although I've only overlapped with a tiny amount of his very long and industrious career. He started his Oxford life at St John's as an undergraduate, did his doctorate, and then spent a little bit of time over in the University of Groningen as postdoc, before he was captured back to Oxford. It's all right, I think it's probably there's a cable underneath. He back to Oxford to do first the junior research fellowship at his old college, and then was kidnapped by Corpus, who offered him a fellowship. That was a very long time ago. 3 years ago, in 2022, he was elected Fellow of the Royal Society, which is an honour not accorded to that many scientists at all, and very few chemists, and also has now retired from teaching, having served his time. So I work with his junior replacement colleague now, and is now able to do actually much more research than ever before. He's a stalwart of the Oxford system, and I'm absolutely delighted to introduce him. He's also written 2 of the clearest books on NMR, one of his main topics. He also studied spin chemistry and the subject of tonight's talk, magnetoreception, which he first got interested in, I think, 20 years ago, and has been the world authority on ever since. Thank you very much.
Thank you, Rachel, and good evening.
Should we turn you down a bit? Do you want to turn this slide down? Try again?
Good evening.
Gosh, isn't that weird? Maybe...
It might be because it's up against your pullover.
I think it might be breathing into the microphone. So if you angle it a bit further away.
I can shout.
No,
Is that any better?
That's better, yeah. Thank you.
Okay, good. Right. So, good evening.
Go on.
I'd like you, for a few minutes, to put yourselves, as it were, in the shoes of a bar-tailed godwit. So a medium-sized wading bird. So you have hatched at the beginning of the summer on the tundra in Alaska, but summer is coming to an end, it's starting to get chilly, and you need to fly south. Winter is coming, and your destination is New Zealand, 11,000 kilometres across the vastness of the Pacific Ocean. But that's not a problem, because you can fly at an average speed of 60 kilometres an hour, day and night, non-stop. All being well, it'll only take you 8 days. But how do you manage not to get lost en route? Well, you can use the sun and the stars to get a sense of direction. If you've been that way before, then your keen sense of smell will help you hone in on New Zealand when you get close. And you can recognise landmarks, but there are precious few of those across the ocean. So what would be really helpful for these long-distance migrants would be a magnetic compass, the ability to detect the direction of the Earth's magnetic field. And remember, the Earth's magnetic field is always day and night, whether you can see the stars or the sun or not, and whatever the weather, it's always there. And as we all know from hiking with a magnetized compass needle, we can use it to get a sense of direction. And it's clear that the birds do too. And what I want to talk to you about is how that magnetic compass might work. So birds don't have GPS, they don't have satellite navigation or magnetometers like we do in our phones these days. They don't have satellite navigation, but they might have chemical navigation. So I want to talk about the possibility that the birds use magnetically sensitive chemical reactions to detect the direction of the Earth's magnetic field. So this is a northern wheatear. These are migratory songbirds. They weigh only about 25 grams. And you can see why a magnetic compass might be useful to these birds. They fly alone and at night without the benefit of experienced individuals to guide them, and they travel huge distances. Baffin Island to West Africa, Alaska to East Africa, and back again, year after year. Even the European robin, so the ones that we see in our gardens are resident all year round, but there are plenty of robins that do migrate. From Scandinavia and northeastern Europe, they fly to the Mediterranean and the Middle East. So the dark blue areas are the resident ones, the light green are breeding birds that will fly to the blue areas in the winter and then back again in the spring. So first of all, how do we know that the birds have a magnetic compass? So this has been suspected since the 19th century and was really only first demonstrated 60 years ago. And we still don't really know how that magnetic compass works. So the kind of experiments that people do are shown with this cartoon. So the birds are tested in what's known as an Emlyn funnel. So this is an orientation cage. It's cylindrically symmetrical. It has a translucent lid, so light can get in, but not see out. So once they're in the cage, the only directional information they have is from the magnetic field that they experience. And if you do this test during the migratory seasons, in the spring or the autumn, when the birds are motivated to fly, then the direction in which they jump in the cages in their attempt to escape is the direction in which they would fly if you release them. So it's a very convenient and well-controlled test experiment to test for migratory orientation. Because all you need to do to prove that the birds are using their magnetic compass is to test them again in a rotated magnetic field and so they jump in a different direction appropriate for that rotation. And then you know that they're using their compass. And the way that you record their directional preference is by lining the sloping walls of this funnel with paper that is sensitive to the scratches that the birds make with their claws in their attempts to get out. And as shown by the cartoon on the right-hand side, this bird evidently was oriented average towards the northeast. So the eminent funnels, here they are, 9 of them. So this is the setup in the laboratory of my collaborator, Henrik Mauritzen, at the University of Oldenburg in northern Germany. He's the biologist that does the behavioral experiments. So 9 funnels at a time, 1 bird in each funnel. The experiment starts at dusk, which is when the birds would normally be calibrated against a compass prior to set to fly through the night. And you put 1 bird in each funnel for an hour. And at the end of the hour, take out the paper and basically count the scratches. So this table with 9 birds in 9 funnels is surrounded by a 2-meter cage of magnetic field coils. So 4 coils in each of the 3 dimensions. And the coils are made of copper wire, and you control the currents that run through those coils very carefully. And the electrons moving within the coils generate a magnetic field. And so this is the way that the magnetic field is controlled, and then by just reprogramming the coils, you can rotate the field direction and see if the birds move in a different direction. And so this is enclosed in a very carefully electromagnetically shielded chamber. You just see the walls of it here on this slide. This is to allow the Earth's magnetic field in, but to keep all electromagnetic interference out. And there are 4 of these chambers in this building on the campus of Oldenburg University. The building itself is made entirely of non-magnetic materials, so as not to interfere with the bird's ability to use their magnetic compass when tested in these emerald funnels. So, the big question is, how does this magnetic compass work? Well, first of all, I'm going to tell you how it doesn't work. So the only animal in which we really understand any kind of response to the Earth's magnetic field is this bacterium. They're called magnetotactic. They were discovered in the 1990s. This is false colour image taken with an electron microscope. This is the animal. It's about 1/millionth of a metre long, and these black blobs are each of them crystals of magnetic iron oxide. And they're not something that the animal has eaten. These are natural. And they act like a compass needle. Each of the crystals is magnetic, and they're lined up so that their magnetic moments reinforce. And this magnet is strong enough that it aligns the animals along the direction of the Earth's magnetic field. Now, you might ask, why does it need to do this? Well, these bacteria live in shallow pools of water, and if they get too close to the surface, then the high oxygen content will kill them. So they need to be able to distinguish up from down as a matter of life and death. And they're too light to use gravity. So they've evolved to use the Earth's magnetic field and to swim either up or down, depending on the magnetic field. And I'm going to show you a video here. So this is taken on a microscope gauge. This is a droplet of water. That's the edge of the droplet. And each of these little specks is one of those bacteria. When I start the video going, you're going to see these things swimming around. And then out of the frame, someone is moving a magnet around. So let's go, here we are. Now the magnet is over on the left-hand side. They're all congregating along the edge of the droplet. Now the magnet is back on this side, and they're all swimming towards me, and in a moment they'll be back again. So this is clearly not a magnetic compass, and you can't imagine a bird would react in that way. It couldn't possibly have enough iron inside it that its body wouldn't act like a compass. So the bird's compass is not based, as far as we know, on particles of magnetic material. It's a rather strange compass. So if you test a bird in the northern hemisphere in the spring, when it's motivated to fly to the north, it experiences the Earth's magnetic field like this arrow. So it has a horizontal component that points north and a vertical component that points down. And if then you test the birds with the horizontal component inverted, so it still points down, but now towards the south, then perhaps not surprisingly, the birds jump in a direction 180 degrees rotated to the south. More surprisingly though, if instead you invert the vertical component, which you would think would have no directional information, the bird still goes south. And it's only when you invert both components, so the arrow points in exactly the opposite direction, only then do they go north again. So this is it's what's known as an axial or inclination compass. And it's not like our magnetized compass needles, which would react only to the horizontal component. The birds are evidently using the vertical component as well. And that's not the only strange thing about the bird's compass. It's light dependent. That's why you needed a translucent lid on the emblem funnels, so you get light in. If you test the birds in blue light, then they can use their magnetic compass perfectly well. Test them under green, yellow, red light, or total darkness, and they jump in random directions. They can't use their compass. So, this led to the idea that it could be a chemical mechanism, which was first suggested in 1978. And so the basic idea is quite simple. So this is a chemical reaction here. We have some reactant which absorbs blue light, and that provides the energy to convert these reactant molecules into short-lived intermediates. And these intermediate molecules can do two things. Either they can come back to the reactants, or they can go forward to form a chemical product, a different molecule. And so all you need for this to be a compass, in principle, is that the probability that it goes forward or back depends on the magnetic field in which you do the reaction, and in particular on its direction. So all the bird would need to do then would just be to move around in the Earth's magnetic field and register the change in how much of this product is formed. And that should contain sufficient information for it to work out where north and south are, and therefore where it should need, which direction it should need to fly. So that's the idea. And so how do you test it? Well, when we started this, as Rachel said, 20 years ago, we wanted molecules. Sorry, this should be a video. For some reason, it's not working. This is supposed to rotate, but it doesn't matter. You'll see a better picture of it in a minute. So we wanted here we are. We wanted a molecule as a proof of principle, because the mechanism I've just outlined, intermediates going forward and backwards, this is a real mechanism. It's well established. It was in its infancy in 1978, but since then there have been hundreds of reactions that have been studied in laboratories that do respond to magnetic fields. So it's a real mechanism, and the question is, could it happen inside a bird? Could it be the basis of a compass sense? And could it be sensitive? Because after all, the Earth's magnetic field is not very strong. It's about 1% of a small fridge magnetic field strength. Okay, so we hit on this molecule. So most people doing laboratory experiments on these kind of reactions would start with magnetic fields, say 100 times stronger than the Earth's, and then work upwards to even stronger fields. We wanted to do the opposite, work down to the Earth's magnetic field strength, realizing that as we took the magnetic field weaker and weaker, the effects would get smaller and smaller. And this is the molecule we chose for this proof of principle experiment. And it's not supposed to resemble anything you would find inside a bird, or indeed anywhere in biology. So CP and F stand for carotenoid, porphyrin, and fullerene. Now, carotenoids do occur widely in biology. They're pigments, and so as the name suggests, it's responsible for the colour of carrots, but also pumpkins, tomatoes, daffodils, shrimps, lobsters, flamingos. These are all carotenoids. Porphyrins, there are compounds like this one in blood, heme groups that are responsible for the red colour of blood. They have this kind of structure, as does chlorophyll in plants, but not strung together like this, and certainly not with this buckyball fullerene on the end. Now, we chose this molecule, of course, because we thought it would have the right chemistry. And so it proved. We were able to show that the subsequent chemical reactions, what happens when this absorbs light is the following. If you choose green light in this case, that's absorbed by the porphyrin, so it becomes energized sufficiently that an electron is ejected from the porphyrin onto the fullerene. And that's swiftly followed by a second electron jumping out of the carotenoid onto the porphyrin. So within a billionth of a second after the photon of light is absorbed, we've transferred an electron from the carotenoid onto the fullerene. Electrons are negative, so we've left a positive charge behind here and put a negatively charged here. And it's this form of the molecule that is magnetically sensitive. We were able to show that its subsequent chemical reactions can be affected by a magnetic field as weak as that of the Earth. And also that if you change the direction of that field, the chemistry also changed. So that established for the first time the principle that you could make a magnetic compass out of a chemical reaction. Okay, now, a little bit more about what we know about the bird's compass, and then we'll get on to the molecule that we think is responsible inside the birds. So we know that the sensors for the magnetic compass are in the bird's eyes, which is perhaps the most obvious place for a light-dependent sensor. Eyes, of course, being responsible for vision. And in particular, we know that the magnetic sensors are in the bird's retinas, so the thin layer of cells at the back of the eye that contains all the machinery necessary for vision. So right at the back of the retina, there's a layer of photoreceptor cells. And this is where the light is absorbed. And then information is transferred from this layer of cells down in various stages through the retina, finally to the ganglion cells, where an electrical pulse is generated and is sent along the optical nerve to the brain. And we think the most likely place for the magnetoreceptors is in one of these types of photoreceptor cells, known as the double cones. And inside those cones, we know that there is a molecule called cryptochrome. And this seems the most likely one to be doing the chemistry I've just outlined. So cryptochrome is a protein. It's quite a lot bigger than the CPF molecule we were just looking at. This is a representation of its three-dimensional structure. So like all proteins, it's a chain of amino acids, in this case about 500 of them. And that chain is arranged in a rather beautiful and complicated shape. This is the backbone, and not shown here, hanging off the backbone, there are the other bits of the amino acids, the side chains, that give it its function. And this slide shows what happens inside this protein when we test it in the laboratory. So the length scale here is 2 nanometers, 2 billionths of a meter. And right in the middle of this protein, there is a compound called FAD, which is related to vitamin B2, riboflavin, and it absorbs blue light, the same kind of wavelengths that the birds need for their compass to work. And what happens when this flavin absorbs a photon of blue light is that, again, it's energized, activated by the energy in the photon, and that causes a series of electrons to jump around within the protein. So first of all, from this group I've labeled A, an electron jumps onto the high energy flavin. That creates a hole in A, which is filled by an electron jumping from B, and then one from C, and then one from D. So this is behaving like a wire, transferring an electron ultimately from D onto the flavin. So the things I've labelled A, B, C, and D, you can see they all have the same shape in green, and that's an amino acid called tryptophan. And there is this chain of 4 of them leading out from the center of the protein out to its surface here. And so these 4 consecutive electron jumps are all over within a billionth of a second. And that forms, like the CPF molecule, it forms a charge-separated state with a positive charge on the terminal tryptophan and a negative charge on the flavin because of the movement of the electron. And again, we've been able to show in the laboratory that the reactions of this state, of this protein, do respond to magnetic fields, and that this molecule could be suitable as a magnetic compass sensor. So, just a couple of words about what we're currently doing, and then I'll finish. So, what next? How do we, it's all very well studying this protein in the laboratory, but the laboratory is not a photoreceptor cell in a bird's eye. We really need to do behavioral experiments to see whether this is the right molecule and the right mechanism. Now, so this is the part of that flavin molecule, the FAD, the yellow compound that absorbs blue light and allows all the electrons to jump around. And once you've added an electron to this molecule, it becomes magnetic, and that's because electrons fundamentally are magnetic particles. But they're not the only magnetic particles in molecules such as this one. So the N's are all nitrogen atoms, the H is a hydrogen atom, and there are various other hydrogen atoms not shown in this representation. And the nuclei of those atoms, the heavy bit in the middle, is magnetic for nitrogen and for hydrogen. So there are internal magnetic interactions which turn out to be very important for this compass sensor, of the electron interacting with the nuclear magnets. And the nuclei have this magnetic property because they're composed of protons and neutrons, each of which are themselves magnetic. But what about the carbons? So all the ones here with blue circles, these are all carbons. And 99% of carbon that occurs naturally is carbon-12, which has 6 protons, 6 neutrons in its nucleus, and it has no magnetism. So it doesn't, the carbon nuclei don't interact with the electrons. But 1% of carbon is carbon-13, which has an extra neutron. Because the neutron has a magnetic moment, carbon-13 nucleus has a magnetic moment. So what if we could replace all the carbon-12 atoms in this molecule with carbon-13, so not 1% natural abundance of this heavy carbon, but 100%, all of it. What if we could do that? Then we would have introduced a whole lot of new internal magnetic interactions. The electrons wouldn't interact only with the nitrogens and the hydrogens, but also with a dozen additional carbons. And that will change the properties of these molecules so that they should respond in different ways to the Earth's magnetic field. Ways that we could detect in the behavioral experiments in the inland farms. And so you might wonder how on earth we're going to do this. How are we going to get carbon-13 into this flavin molecule, into cryptochrome, into the photoreceptor cells in the bird's eyes? Well, we haven't done it yet, but the way we think we can do it is as follows. So birds, like humans, don't make their own flavins. We need to eat riboflavin as a vitamin. If we don't, we will get ill. If you look on your cornflakes packet, you can see it's enriched in riboflavin. It's an essential molecule. Birds are the same. If you give them a diet with no riboflavin, within 2 weeks they get very ill. If you continue, then they'll die shortly afterwards. So it's an essential molecule and the turnover is quite high. So we hope we can replace all the flavins in their body, not just in their eyes, with carbon-13 riboflavin. So we're going to start with glucose, just a sugar. You can buy this. And it has carbon-13 in all 6 positions here. We will then feed that to a modified form of yeast, which will then make riboflavin, this is the structure here, with carbon-13 everywhere in it. It's a yellow powder. Now, we can't feed that to the birds. They won't eat a powder of this sort. So we feed it first to mealworms, which the birds love. So the mealworms need to have a very carefully controlled diet. First of all, they must not have any riboflavin that has carbon-12 in it. So we need to deplete their normal diet remove all the carbon-12 riboflavin, so we can put this carbon-13 one in its place. And we have also to make sure that the mealworms have all the other nutrients that the birds need to thrive. And then you feed the mealworms to the birds. The birds are quite happy to eat only mealworms. And we're at the stage now that we can get the carbon-13 riboflavin very efficiently into the mealworms. The birds will eat it, they stay healthy. As far as we can tell from analysing their droppings, they are incorporating it into their bodies. And we're in the middle of the migratory season in the spring now, and so these birds in Oldenburg are being tested to see if they orient in a different way, a different direction, better, worse, than they would normally do. And in that way, we hope to prove that cryptochrome is the right molecule, and we've got the right mechanism as well. Okay, so let's wind up now. The question I'm often asked is, how do the birds actually perceive the Earth's magnetic field? Well, we don't know, but as I've said, the sensors are in the retina, so it's possible that magnetoreception just piggybacks the visual reception system. So at night, the cone cells in the retina are not very active. They're colour sensors, and birds like us can't see colour at night. So maybe the magnetic sensing takes over the cone cells, and the birds literally see some form of the Earth's magnetic field. Okay, so we mocked up this thing. We took a drone out into the countryside, close to where I live, north of the city. We strung a video camera below it, and we flew it around in a circle. And then we took the footage and we superimposed it, the pattern that we thought the bird might get from a cryptochrome sensor. So this is pure science fiction, don't take it too literally, but just to give some idea of what it is the birds might perceive. So here's a compass rose in the top right corner, and that shows which way the birds are looking. And the blue and the pink things are the calculations. So as the bird comes round, it's looking due north now, and this cross shape is right in the middle of the field of vision. And that could tell the bird that that's the north-south axis. So just fantasy really, but it must be something like that. The bird must have some perception of the Earth's magnetic field. Okay, so what use could all this be? Well, I mean, my fundamental interest is to solve this problem. It's one of the few sensory mechanisms in the animal kingdom that we don't know much about. We know a huge amount about vision and taste and hearing and all the rest, senses that we have. We don't have a magnetic sense, and that makes it more intriguing, perhaps, to think about how animals actually do it. But it would be nice also if there was some application of all of this. The displays in your mobile phones these days, almost all of them constructed from organic light-emitting diodes. And it turns out that the physics of these OLEDs is very similar to the chemistry I've been talking about today. So the molecules that I've shown you that respond to magnetic fields are analogous to the so-called electron-hole pairs formed inside these organic semiconductors. So not exactly the ones in your phone, so you won't see anything interesting if you wave a magnet near your phone, but related molecules show magnetic field effects, have basically the same physics. So this opens up the possibility of making, as it says at the bottom, cheap, disposable, non-toxic, biodegradable devices that we could use for sensing the presence, direction, strength of weak magnetic fields. So not using heavy metals, which are expensive and toxic, but just using cheap materials, and then getting organic chemists to make, like Rachel, to make the right molecules with exactly the right properties to show magnetic field effects. And the other potential application is conservation. So populations of migratory birds are falling, partly as a result of global warming, partly habitat destruction. And one of the ways of dealing with threatened species of birds is just to move them, translocate them to somewhere safer. But if they're migratory birds, they just fly back again. They're such good navigators. So we need to know how to fool them to stay put in a safer environment long enough that they can breed and then their offspring migrate and come back to the safe location. And we can't do any of that unless we really understand how they navigate and how they know where to return to after flying thousands of kilometers there and back. Okay, so of course I haven't done all this on my own. There have been large numbers of very talented students and postdoctoral workers over the years, too many to list here. I just want to mention the names of my main senior collaborators in Oxford and in Oldenburg, and the grant we currently have from the European Research Council that is funding all of this. So I want to thank all of them, and thank you for turning up this evening and for listening.
Thank you, Peter. I've actually never listened to his talk before. I know obviously the kind of work he's been doing, but it's a real treat to listen to it from kind of start to finish. The public version. There's a bit more chemistry hidden behind it. We're going to take a very short break now, a short refill break. We'll see you in a moment for the discussion. Thank you. Right. Okay. Right. Just to explain how the next bit goes, we've got people in the room, we've got people online. Susanna's very kindly going to do questions that are online, going into the chat. Feel free to merge them if you've got several all very similar. Okay, we don't need to go through necessarily in order. And if you want to catch my attention, just basically look terribly alert and intelligent, and I'll, like you want to say something, I'll come over. When you are using this microphone, please make sure it points directly at your mouth, not down there, that doesn't work. So you actually need to point it at your mouth like a superstar, pop star, and then it works absolutely beautifully. We have this lovely handle which a former regular at the cafe made for us to make sure you point it at your mouth like that. And we'll just come round. Does anybody want to kick off? Yes.
Yes, how did they know to go east and west as well as north and south?
Yeah, they have, the birds are born with
Is the
inherited...
microphone on?
I think
Is it
so.
on? Sorry. Try that? Yeah, that'll be
That's
it.
better, sorry.
Thank you.
They inherit instructions, in effect, so that the birds know, for example, that they need to fly southwest for 2 weeks, 1 week southeast, and that will take them from Scandinavia to North Africa. So this is something they inherit from their parents. And then they use their compass to work out where southwest and southeast are.
What happens then if a bird is very slow at flying?
It's very...
Slow at flying, and its 2 weeks don't take it far enough.
Well, sorry, perhaps I oversimplified it, the 2 weeks. It's not done entirely, but... So, for example, flying from Scandinavia to North Africa, the birds don't like flying over open water, so their program tells them either to go via Gibraltar or at the other end of the Mediterranean. So, they'll be alert to the countryside, the appearance of water, and so on. And they'll have other cues programmed that will tell us where they need to change direction.
So I'm just going to come to 2 people in the room and we'll go online for a bit.
Thank you. I'm not a chemist and I'm not an ornithologist either. So forgive the slight ignorance of some of these questions. But my first question regards the chemical reaction that you outlined about the jumping of the electrons, etc.
Yes.
That was initiated by blue light.
Yes.
What happens at night?
Yeah, it's a good question. So it's never totally dark at night. I mean, if it were, the birds wouldn't be able to see anything, and they'd probably just go to sleep rather than attempt to fly large distances. So they typically fly these small songbirds above the cloud level. So they always have starlight. They don't need the moon, and they don't need the sun, although the sun can be useful at dusk to calibrate the compass and get them off in the right direction. So there must be enough, if this is the right mechanism, there must be enough light in starlight to allow them to do this. Yeah, I can see that you're sceptical.
Yes.
And I agree with you.
A little bit sceptical, yeah. But next question. You were talking about the mealworms and injecting the mealworms, which is fine, but you can't follow the birds with a packet of mealworms. They're going to land and eat something else.
Yeah, I mean, they're all going to be tested inside these Emlyn funnels. So they
right.
can be tested
this
in
is
the
not
lab.
this is not a real life test, no.
No. So, I mean, that's, of course, you're right, much more challenging. I mean, what can be done is to see which way they set off, whether they set off promptly at dusk, at the normal time, or if they're delayed by either treatment with different magnetic fields, or we could do it with the C13 mealworms. And it is possible to track the birds over some distance. So there's an ornithological station on Helgoland in the North Sea, and then an array of receivers along the northern coast of Germany and the Netherlands. So you can track the birds from the island and work out where they hit the land. But yeah, it's much more difficult because they have all sorts of other information there to navigate by.
Yeah, a number of questions. Those photoreceptors they use, double cones, are they related to the ones that we use for knowing whether it's day or night?
So yeah, I mean, we have rods and cones.
And then the third ones? And then the third ones that we use to tell whether it's day or night, or what time of day it is?
Oh, for the circadian clock?
Yes.
No, so they use different photoreceptors, and they're not in that outer layer of the retina. So they have melanopsin.
What the ones we've got?
That's right. I mean, the birds probably have the same. No. So I mean, the cones are all involved in vision. I mean, the double cones are attractive because we know the cryptochromes are there. I didn't say the birds have 6 different cryptochromes. We think we know which one is most likely. And it is in the double cones and also the long wavelength cones. And the other reason why the photoreceptors are an attractive location is that because you want this to be a direction sensor, the proteins must be aligned relative to the bird's body. So if they're rotating or just randomly oriented, then you could have an intensity sensor, but it wouldn't give you any directional information. And the structure of the photoreceptor cells contains ordered membranes to which the cryptochromes could be attached. That could be a way of aligning them.
Because they all sit radially. Because all the cells all sit radially.
Exactly so. They're all aligned. Yeah, indeed.
And the other thing was, since they're using dead reckoning, then does that restrict, if you're trying to persuade them that you want to move them to somewhere else, is the geometry of their route, will that be limited? Because if they're not particularly investigated, dead reckoning, you may have a super site for them to go off to, but their navigation means they can't actually do that geometry.
You just quickly summarise that, so it didn't come through. Can you repeat the question just briefly?
Okay, so if I understood the question is about translocation of the birds?
Yes, in translate.
Okay, so that has been done, both physical translocation but also virtual translocation by choosing the currents through the coils in the Emlyn funnel experiments. You can fool the birds that they're actually in Russia when they're in fact in Germany, and then see which way they go. So they can do true navigation. So as well as having a compass sense, they have a magnetic intensity sensor, which amongst other things will allow them to work out where they are on the surface of the Earth. So if you translocate them, then they know to go in a different direction to get where they want to go. I mean, if they didn't have that intensity sensor and they were programmed to go southwest, when you took them to Russia, they would still go southwest, which would be an inappropriate direction. But because they can work out where they are, they compensate for that. Okay,
I don't think he's convinced, but we're going to carry on. So Suzanne, do you want to read out some from the chat?
Hello. I will read them out as they've been written. There's one from Dave. that says, the Earth's magnetic field flips occasionally. Will that flip mess up their navigation completely?
Probably. It doesn't happen very often. If it happened instantaneously, the birds wouldn't know about it because their compass is not sensitive to exact inversion of the magnetic field. It actually takes the order of 10,000 years for the reversal, and during that time the magnetic field will be locally much weaker than it normally is. However, because it happens relatively slowly, it's possible the birds can adapt and still use their magnetic compass during these reversals. The alternative is that they simply ignore the magnetic information because the field is unstable and then rely on other directional cues, and we don't know which. We are due for another reversal soon, sometime in the next 100,000 years, probably.
Soonish. Do you want to carry on for this?
Another from Marion Bird. Do birds which don't migrate have cryptochrome inside the double cones in their retinas?
Yes, they do. So this mechanism could be common to all birds. It's more difficult to do the testing on birds that don't migrate because they don't display this migratory restlessness behavior that causes them to jump inside the emblem funnels. So for non-migratory birds, you have to use conditioning experiments, get them to associate a magnetic field with a reward, food usually, and then test them without the reward and see if they can still recognize the magnetic field. And some people, they can claim they can do that even with domestic chicken. Other people have failed to be able to do that. But it seems likely that all birds probably have this ability. They may not need it, and they may not use it.
So I had a question from right at the very beginning. You talked a bit about calibration. Why would they need to? Is it the kind of thing where when you haven't used it for a few hours, you're not going to get it right? What's going on with calibration?
Yeah,
Do you know anything about how it might happen?
There's not a great deal known. No, I think it's maybe, as you say, it just reminds them of how they should use this information. I mean, the sun is setting, even when it's below the horizon, you can still see which direction the sun had been. And so that's useful information, so why not use it? And if it helps extract information from the magnetic compass sense, then that's something that presumably was optimized by evolution.
The other thing I was thinking of was the fact that insects can see ultraviolet lights. That's another sense we don't have. We have vision, but not in that range, so we think things are different colours from them. And I was thinking about, it must have been a talk on bees at the cafe, probably talking about, you know, their various ways they do things, and they have a kind of orientation moment when they come out of the hive, when they're kind of dancing in the air to get their bearings. Is it possible that anything like insects would have this? Does it have to be about migration? Could you use it for shorter term navigation? Could bats have it, for instance? I know they use echolocation, but they also need to know where they're going. They fly quite long distances.
There's much less known about animals that are not migratory birds. So the cryptochromes also occur in fish and amphibians, which seem to have some kind of magnetic sense. So that may be a common mechanism to the birds. Other animals have a polarity compass, like a magnetized compass needle, so that suggests magnetic iron oxide particles, and that probably includes insects and the bats as well. So other animals have a magnetic compass which is not light dependent, so that again makes it a bit less likely to be the mechanism I've been talking about. We did at one point, just very briefly, an obvious experiment would be to produce a migratory bird with no cryptochromes and then see if it could orient. Now, that's not likely to be possible for a variety of reasons. One of which is that you can't breed these wild birds in captivity. So you can't do the kind of genetics that are possible with fruit flies. But even if you could prevent cryptochrome being formed specifically in the bird's eyes, this wouldn't tell you that you'd identified the right molecule, because cryptochromes do all sorts of things. Their major function in humans as well as birds, plants, insects, is regulation of our circadian clock. So it's possible that cryptochromes are involved in magnetoreception, but not as the sensors. Maybe they're involved in transmitting that information downstream between the sensor and the brain. So if you could produce a bird without cryptochrome and it didn't orient, you wouldn't know whether you'd disabled the sensor or something else that is essential for magnetoreception. So we thought that fruit flies, Drosophila, would be an ideal model organism because then you can do the genetics. You can produce flies without cryptochrome. Or you can take their cryptochrome out and put a different one in, human one, for example. Cut a very long story short, we tested 110,000 flies and we couldn't find any magnetic field effect on their behavior, despite the fact that there were high-profile reports in the scientific literature that fruit flies do use magnetic fields. So, sorry, this is rather rambling and long answer to an earlier question, but it is possible that animals that don't travel great distances may have a use for the Earth's magnetic field. And so that might include flies, insects.
Thank you.
You showed the bar-tailed godwit.
Yes.
I just wondered what percentage of these birds that are flying south actually make it to New Zealand. Is that known?
Yeah.
How many get lost?
So the study that tracked these birds, I think, started off with about 20 with radio tags. That's how they were monitored. And some of them, the tags fell off, and so they don't know what happened to the birds. Others were blown off course by storms, but to some extent they can compensate for that, as we were talking about earlier, because they can work out where they've been blown to and then compensate to try and reach. So I think something like a third of the birds tested actually got to New Zealand in a reasonably direct route. Yeah, it's a hazardous business, migration. The small songbirds, something like 40% don't survive the first journey, but if they've done it once, then they have a much better chance of getting back again.
Are there no islands? Are there any islands on the way from Alaska to New Zealand that they can...
Sorry, islands?
Islands.
Places to stop.
Fiji?
Oh, yeah, sorry, islands, yeah. Well, I guess there's Hawaii in the middle, but I don't think there's any evidence that they stop there. I mean, the godwits don't stop on the sea. They have to keep flying. They don't glide, so they're flapping their wings the whole way. Yeah, I mean, you'd think that it's not an awful place to go.
You just over the cable, I think.
Oh, sorry.
No, it was you.
So, yeah.
Adding to what Rachel was just saying. Adding to what Rachel.
I'm sorry, guys. Might be a little battery problem, just give me a sec. Always possibility.
how do they know about crosswind? How do they measure the crosswind? Otherwise, with deck reckoning, they will still be off.
Okay, let's go here, please.
I guess again, it's, you need to be able to locate yourself. I mean, just like a map and compass.
Yes.
So,
With a crosswind with no features. That's why Second World War bombers used to go and bomb the wrong city.
okay, but if you have some mechanism of working out where you are, then presumably you can compensate.
Okay, back this slide, please.
Yes.
Just adding to what Rachel was saying, if we think of magnetism as like a sixth sense, and you've proved with those experiments that varying the magnetic field has an impact on the bird's orientation, could there be a seventh or indeed an eighth sense involved?
Ah, gosh. Well, in birds, I'm not sure. Other animals, maybe. So there are shrimps that can detect polarized light. So, like sunglasses will filter out reflected light using its polarization. So that this is a defense mechanism, so that underwater there are lots of light reflections, and you can more easily detect predators if you're a shrimp, if you can detect the polarization of the light that is reflected off the animal that is wanting to eat you. So these shrimps have developed this, which I guess you might call an additional sense. I mean, it's not something that humans can do. Birds may be able to, we're not sure yet. But then the predators, it's kind of an arms race, then they learned to do this as well, so that helped them locate the shrimps. And then the shrimps have to find some other way of spotting the predators, which this predator's not yet wise to. So shrimps, we have 3 different colour channels, and then the cones for night vision. Birds have 4 different colour channels and the double cones, and the single cones as well. These shrimps, if I remember, have 12 or 13 different visual channels for different colours of light, UV as well as visible, and all these different light polarizations. So maybe that's kind of 6th, 7th, and more senses.
One I was wondering specifically in terms of migratory birds, they might be using a sense which we haven't yet discovered.
I can't think of anything more than I just said. Let's see. Do you have something in mind? No, I don't think. I'm not sure there is.
Focused in on.
Yes. Yeah.
Science is very specialised nowadays. We're just going to go online because there are lots of good points.
Sorry, there's lots of questions online. So from Chris, we were taught about the Coriolis component effect. Are there different mechanisms for different birds?
Probably not. Well, there's one exception. So all of these migratory passerines, the group that includes the robins, the wheatears, blackcaps, the other ones, the pictures I've been shown, they all have cryptochrome. There is an American New World woodpecker that doesn't have cryptochrome and does migrate. And so this is being studied at the moment to see whether it has a different mechanism. So that's an interesting, naturally occurring control group. But for all the others, for which the genome has been sequenced, they all have this cryptochrome 4 and probably all have the same mechanism.
From Steve, in an early slide, you showed that it's the orientation of the field line that is important, not which end is north and which end is south. Does that play with it affecting a reaction? I would have thought that the sense of the field would be important. I'm not really sure what that is.
So I think he means directionally or something.
Yes.
Yeah. Does that make sense to you, Peter?
Not much, but so I mean, one feature of this that the birds are sensitive to the inclination of the field, so they're not actually distinguishing north from south. They're distinguishing towards the equator or towards the pole. So that's subtly different kind of information they get from this inclination compass that you wouldn't get from magnetic iron oxide particles.
From Caroline. Caroline, I'm not sure how to pronounce her name.
Speak up a little bit, will you?
Sorry. From Caroline. I heard a talk a while ago by Jim Al-Khalili and John Joe McFadden, in which they said the mechanism for robin migration was quantum mechanics. I couldn't really understand it, but they wrote a book about it. Would this mechanism be compatible with your proposed mechanism? If so, how would that work?
Okay, it's exactly the mechanism they were talking about in that book. Yes, I've avoided talking about the detailed mechanism, but maybe I can just show you one extra slide if this thing will work. No, it won't. It is a fundamentally quantum mechanism. You may have noticed this on the final slide. This Quantum Birds is what we called the research grant that's financed all of this. The slide I wanted, if I can find it quickly. Okay, so this is a very simple example in which I will try to explain the mechanism in a little bit more detail. So I'll try and do that quite quickly. So two very simple molecules. These are called methyl radicals. Radical means that these molecules have an odd number of electrons. Most molecules have an even number of electrons, and that means basically they're non-magnetic. But you can form molecules. They're usually very reactive, high-energy molecules, like the methyl radicals, which have an odd number of electrons. And the dot here is supposed to indicate that odd electron. And molecules like radicals that have odd electrons but can't pair up with any other electron, these are magnetic. Now, so if you imagine you could form some of these highly reactive methyl radicals, and they just float around in water, say, as soon as they bump into one another, they will react and recombine to form a much more stable molecule, and that's, in this case, ethane. But it's more subtle than that. When you form that bond between the two carbon atoms in the methane molecule here, that's formed of two electrons. Electrons have a property called spin. This year is the 100th anniversary of spin. It was first proposed in 1925. And the spins of the two electrons in this bond are represented by those little red arrows. And the Pauli principle, which is a fundamentally quantum principle, tells us that those two electrons must have opposite spins, one up and one down. And when the two methyl radicals combine, they do so without a change in their spins of their electrons. And so this can only happen if the two methyl radicals collide with opposite spins, then they can recombine. But of course they don't have to encounter with opposite spins. They could have the same spin. And quantum mechanics tells us that there are only two options. It's either one up, one down, or both the same. So if this happens, then they can't react to form ethane, because that is not allowed. The reaction would not flip over one of the spins, and so you can't form a molecule that has two opposite spins. So these have to do something else. And if this is done in a liquid, they will react with the solvent or some other molecule you put in there deliberately, and they'll form a different reaction product. So we've got two different reactions happening in parallel, just depending on the spins of these two electrons.
Dan, thanks, I enjoyed that.
But
We're nearly finished.
they're not independent.
No,
They can be integrated. So if you think about a compass needle,
no, it's just
how would,
answering some questions.
Sorry, don't worry, good.
how would you get the compass needle to flip over? Well, you'd wave a magnet near it. And so you can change a singlet pair of radicals into a triplet and vice versa by applying a magnetic field. That will just flip over one of these spins, convert the singlet into triplet, and therefore make this reactive. So I think you can see that there's a competition between these two pathways that is regulated by the magnetic field in which you do the reaction. Okay, so by the time all the radicals have recombined, reacted, the amount you get of each of the products depends on the magnetic field. And the quantum bit is that this bit here is not a regular chemical reaction, but it's an oscillatory behavior. The singlet converts into triplet and back again, round and around, millions of times per second. This is a quantum mechanical behavior. So the quantum mechanics is fundamentally at the heart of this, and that's why such weak magnetic interactions can affect these types of chemical reactions. I mean, normally, you just calculate the energy of interaction of the Earth's magnetic field, which is pretty weak, with the magnetic field, magnetic moment of an electron, which is also pretty small. You find that the energy is a million times too small to affect the chemistry at all. Except for this very special mechanism, which gets around that kind of thermodynamic argument. It's irrelevant. The quantum mechanics can be affected by very weak magnetic interactions. And that's, we believe, the origin of this compass, and where the quantum mechanics comes in.
So do you have radical pairs inside the cytochrome then?
Yes. Indeed, formed by the electron transfer reactions.
Okay, with a bit more time. Oh, yes.
Another question by Dave. Do monarch butterflies have a similar mechanism?
Maybe, yes. We don't know for sure. That could be magnetite. For sure they travel large distances. They do have a magnetic sense. It's not yet clear what the mechanism is.
From Paul, who says, I may have missed it, but are the maps passed down from parents via genetics, or are they learned by following the elders in the pack?
So the compass sense is passed down genetically. The map sense is something they have to learn. So this is part of the reason why they have a much lower success rate the first journey. They need to learn the magnetic map information. So how the strength of the magnetic field varies with position on the surface of the Earth. So that's learned.
From Paul, do birds take an exact return route, or is it sometimes via different waypoints on the way back?
They seem to come back by a broadly similar route. I mean, it's not grossly different. I mean, the ones that go from Scandinavia to North Africa via Gibraltar will come back via Gibraltar rather than taking a detour via Israel. So not exactly, but they can come back, if they're lucky, year after year to the same branch each time. So I mean, once they've been there once with their sense of smell, recognizing landmarks, they can come back with centimetre precision.
I think two more here. So, Paul, will location chemical indicators such as smell be used for the final homing?
Yeah, but again, if they've been there before, so the familiar odours. So the seabirds in particular, an enormous fraction of their brains are involved in sense of smell. I mean, detecting shoals of fish at enormous distances is something they can do. And so that probably, the sense of smell there probably overrides the magnetic sense in many cases.
The last one from Jonathan, which is quite witty, so I'll read it out as it's been written. How does Professor Hore visualise the expression bird's eye view? Before tonight I thought it was a top-down perspective, but now?
Well, we don't know what the birds perceive. It's fascinating to speculate. I showed you the science fiction video. The brain's way about a gram. And I mean, the expression bird brain, I mean, I think you should regard that as a compliment rather than an insult. These birds are not stupid.
Just before we finish, would you like to show everyone your favourite slide?
There we are. I'm a physical chemist. Thank you. You've been a great. Thank you for the question.
Thank you so much, Peter. Absolute tour de force. Just before we wrap up, our usual thanks to Pharmacology for hosting us, because it's amazing they let us have this space. To everybody online, to everybody in the room, thank you for coming. It makes it so much nicer for the speaker if he's got somebody to talk to. Thank you very much. And please do join the mailing list if you haven't. I will get this and the last video up quite quickly, if that works. Really fantastic evening. Thank you, Peter. It's been well worth it. So I waited till he's retired before asking him to do it. I've been meaning to ask for years, but this is absolutely perfect. Thank you very much, everybody, and thank you again, Peter.